Hello, everyone, and thank you for joining us for this How Can Plant Tissue Testing Improve Your Crop Performance webinar. My name is Catherine Garcia, and I will be the moderator for the webinar. As a reminder, this presentation can be used for CCA continuing education credits using the QR code at the end of our webinar. In 1959, AgSource was formed to serve Wisconsin dairy farmers. What began as a regional dairy records processing cooperative has now grown into the AgSource of today, serving industries that span the entire agricultural ecosystem, from cutting-edge soil, plant tissue, and water testing to agronomic consulting and full-service DHI testing. AgSource at its core is a source of truth. One test at a time, we help our clients make the best decisions, always grounded in data. I'm now excited to introduce our speakers. Our first speaker of the day is Dr. Jim Friedericks. Jim holds a position of Outreach and Education Advisor for AgSource Laboratories, coordinating the education programs of AgSource and supplying technical information to industry agronomic clients throughout the country. Soils and environmental sciences are central interests in Jim's work and educational experience. He received a BS in agronomy from University of Kentucky and a PhD in agronomy focused on soil fertility from Virginia Tech. Jim has over 25 years of experience in soil testing, joining the AgSource team in 2012. Also presenting today is Garret Johns, an agronomic support specialist at AgSource. Garret has over 10 years of agronomic experience. 7 of those years were spent with a large ag retailer where he served as a precision crop manager and helping facilitate weekly tissue sampling program for corn and soybeans. He currently assists the industry and agronomic clients throughout the world with questions pertaining to agronomic testing. He received a BS in agronomy from the University of Nebraska-Lincoln in 2016 and joined the AgSource team in 2021. I'd now like to turn it over to Jim to get us started.
Thanks, Catherine, for the introductions, and welcome to our webinar today. Today, as Catherine said, we're going to talk about plant tissue analysis and explore some of the areas in which we can use plant tissue analysis in understanding our crop performance. And so we'll be asking, the question, how can this help improve our crop performance? So as we get started, first a little bit more about the laboratories in general. AgSource Laboratories Company has 4 agronomic labs, one in Lincoln, Nebraska, another one in Ellsworth, Iowa, and 2 in northern Wisconsin, one in Bondwell and in Stratford. All these laboratories focus on agronomic soil testing, which includes soil analysis, plant tissue analysis, and in a couple of cases, manure analysis, as well as some water analysis. Excuse me. The other 2 labs in Wisconsin, the Menominee Lab is a large dairy milk testing lab for the dairy industry, and in Marshfield they do food testing as well as water and wastewater analysis that serves the industry and rural populations in Wisconsin. Excuse me, a bit of a frog there, sorry. Peter Drucker is a smart man, and I'm sure he may know something about plants and plant tissue, but I'm not sure he's an expert. Where he is an expert is in terms of management. And his maxim that if you don't measure something, then you can't manage it, applies very well to what we're doing, what we want to talk about today, and what we're doing in plant tissue analysis. So next slide. The purpose of plant tissue analysis or plant tissue testing is to measure the concentration of nutrients that occur in the plant, and then make inferences from that in terms of the health of the plant and the ability of the crop to take up the nutrients that we apply. So it may be used as a way of checking a visual symptom. If you see something going on in the crop or in the plant that you're growing, and you think maybe something's wrong with it, it's a way of double checking that to verify what you think you see if it's actually true. It also helps in understanding how nutrients that are available in the soil get into the plant as it's growing, or is it taking up the nutrients that we think it is, in the amounts and at the time that we think it's doing that. And then also specifically, we can look at treatments that we apply to the soil and see if that treatment is meeting a need that we're trying to address by applying a nutrient to the soil, or if we're applying it to the plant and seeing how it's moving within the plant. So it's a way of analyzing concentrations in tissue parts of the plant, whether it's a tree or perennial grasses or whatever it might be, and then assigning understanding of those concentrations to our understanding of what the crop is expecting and requiring for its good growth. So as a way of managing plant growth, as I said, it's a way of identifying nutrient deficiencies. It can be used in troubleshooting. Visual symptoms are easy to identify and confirm, but it's also possible that there are symptoms that we can't see, or there is a deficiency that we can't see visually. And so an analysis of the tissue helps us to identify those and address those before they become a visual problem. Also, it's useful in a way to look at a crop, and you might discern that part of the field is doing better than another part of the field. And so you want to understand what's going on between this good area and the bad area. And so taking corresponding tissue samples from those areas is one of the tools that we can use then to discern what's going on. It's also possible, useful in terms of adding more information to that, what's going on in the soil. We should include a soil test analysis or even a physical examination of the roots of the crop to see if there's any other impediments to the growth. But tissue analysis gives you a lot of information that will help you answer those questions. It's also a way to monitor nutrient management, nutrient uptake across the growing season. Are we applying enough nutrients to meet the need of that growing crop? Or possibly, is there an excess amount of nutrients? And you can't tell that visually, but through an analysis of the tissue, you can see that there's an excess amount, which means there's more than what's required to meet the crop requirements. And this is something that we need to be aware of so that we can manage more efficiently and economically the nutrients that we're applying to the crops. Then, of course, as we manage those nutrients, we want to be able to evaluate what effect our practices are having on the crop itself. Are we applying the right source of nutrients? Is it in a rate and at a time that it's useful for the crop when we apply it? And are we putting it in the right place? It may be just broadcasting on the surface of the soil is not getting into the soil enough to meet the needs of the crop. And so maybe there's an application that needs to be changed in order to meet those needs. So those are good ways of evaluating the treatments, the applications that we're making, but also as we analyze that, it's useful to look at that season-long application process. Sometimes we apply nutrients before the crop is planted or at planting, and other times we come back after the crop is growing and add more nutrients. And so it's a way of evaluating those different applications and making sure that they're being available and being utilized by the crop as we go through the growing season. Other times, and some particularly for micronutrients, you can apply nutrients to the leaf as a foliar application and then monitor the crop to see if the application from the older parts of the plant, the mature leaves, is actually getting translocated to the growing points, to the point where the seeds are being formed, et cetera, so that we know that we're getting enough tissue translocation of that nutrient to take care of those needs as well. Sometimes also adjuvants are added to these foliar applications, which make it more easily absorbed or makes the material wet more easily and efficiently on the leaf so that it's taken up more easily. And so it's useful to evaluate the effectiveness of that adjuvant to see if it really did improve the application of that foliar application or not. Just to summarize then, there's 2 or 3 different ways of using plant tissue analysis. One is an observational and diagnostic tool, confirming symptoms, addressing deficiencies that you think you see or that you confirm from a measurement, and then also just evaluating the growth of the crop and making sure that all the areas are getting everything they need so that there's a good response throughout the field that we're trying to produce on. And then using it, as Drucker would say, as a management tool by measuring something, measuring the variability of the nutrient concentration in the tissue, and then keeping track of that through the growing season in response to the treatments that we've applied, and just observing how those trends take place so that we know and understand nutrient uptake and nutrient use, as well as knowing and making good judgment on when's the right time to apply those nutrients for the maximum efficiency. Keep in mind that all these uses so far, we're just looking at the concentration of nutrients in the tissue. So is it 5% or 4% nitrogen or above or below that? So it's just a percentage. But really what's happening in the field is there's an uptake of nutrients from the soil or the application to the tissue, and it's a concentration that's spread out over the whole biomass of what's growing in the field or in the plant. And so that total nutrient usage is a concept to keep in the back of your mind, just to realize that everything that we're talking about in terms of percentages has to be considered in terms of the total biomass that is growing in that field. And that is particularly important in situations where you're trying to preserve that nutrient concentration, as we would do with a cover crop. So we have, at AgSource, we have a cover crop biomass assay, where we take a small sample of the total biomass that's growing in the field, cut that at surface level, weigh that green material, and then take a subsample of that green material and send it to the lab for analysis. That way, with the analysis of the dry material, we get a concentration, and then we have the total biomass on the other hand, so we can figure out how much nutrient is being taken up by the growing cover crop and how much of that will then potentially be recycled back to the crop as it's decomposing and being recycled for the next crop in the cropping rotation. Thank you. All of this, as I just indicated with the biomass sample, all of it relies on collecting a good sample. And the quality of the sample that's collected is directly related to the quality of the results that we'll get back after the analysis. So it's important to collect the correct part of the plant. At different growth stages, each plant has different parts that are important to sample. In a corn plant, for instance, when it's small, you want to sample the whole plant up to about 6 or 12 inches tall. But as the corn matures, different leaves are the more important part to sample, and there's usually guides that we have that will guide you through that. Not only do you want to collect the right part, but you also want to collect enough sample so that it's representative of the area that you're sampling. So go through the area and collect 10 or 20 or so leaves or samples from that area. Combine them into one large sample, mix that really well, and then subsample that and send that into the laboratory. For a sample with a lot of leaf tissue, such as corn plants, we would only require about what would be about a softball size ball of tissue. That's not really wadded up, but just, a loosely held softball size of tissue. Keep in mind, if you're sampling petioles in a potato plant, it's a number of petioles, maybe 30 or 40 or 50 petioles, but it's a very small amount compared to the total that we would get off a corn tissue sample. Next slide. These requirements are specific to different crops, to their different growth stages. We have that information on our website. Any laboratory that you use should be able to provide you this kind of information. And so look for that on the websites of your laboratory and follow those guidelines carefully. Time of sampling, the right sampling part, and collecting enough sample. Those are the critical things that are important in the field to get a good result from your analysis. As I said, the quality of the results depends on the sample that is submitted to the laboratory. And so it's up to the sampler to do that. When you do send in a sample, make sure that you sample it early in the day if possible. Keep it cool and mail them in that same day if it's possible, so that the lab gets them as the next day or the next couple of days. Also, it's good advice to sample early in the week so that the sample arrives before the weekend, so that the laboratory is able to process that and it's not sitting in a hot mailbox somewhere decomposing over the weekend. When the laboratory gets that sample, it'll dry the whole sample, grind it up, and then take a subsample of that dry ground material, and that's what's used for the analysis, for digestion, for extraction, whatever the process is, for determining the nutrient concentrations. One thing to keep in mind if you're sampling and the samples are close to the soil surface, or lower in the plant, it's a small plant and there's dirt that's splashed up on it, it may be necessary to wash that tissue just to remove the dirt. Or if there's fertilizer or residue on the leaf from a foliar application, you might need to rinse the leaves quickly, use pure water for that, and then blot the leaf dry, make sure it's not really soggy wet when you pack it up, and then send it in to the laboratory for analysis. All those things are the steps that we take to make sure that in the laboratory, we can get a good result from the analysis. So drying the whole sample, mixing it well, and then subsampling that helps us to take out that randomness of the variability of different leaf concentrations that may be mixed up in that sample. So if you look at this figure, the lower right, if we didn't do a good job mixing that sample, we'd have those values kind of spread all over that. What we're trying to do is minimize that kind of variability by making sure we do a good job processing the sample and then subsampling that for the analysis. And the objective is to improve the accuracy there on the left axis and getting a closer distribution towards the middle of that circle. On the right side is a precision measurement, and we try to maximize precision, but that's done primarily through optimizing the instruments that we use for the analysis. So by carefully calibrating the instrument, we can make sure that we can repeat that analysis very well every time we do the analysis. But if our calibrations are off, you can see that there might be a bias that's introduced, and we may be able to reproduce that value, but it may be wrong because of that uncertainty of that bias. So those are the things that we do as a laboratory to try to improve the accuracy as well as the precision of our work so that we give you the best results when it comes back. Part of that maintenance of that accuracy and precision is by joining a proficiency program. There are several programs in the country. 3 of those are shown there on the right. We participate in all of those. The ALP and the NAPT send samples to the laboratory. And those samples we know that we're getting them, but we don't know what the values are. So that's why we call it a single blind analysis. We run those results 3 or 4 times a year, depending on the program, send the results back to the program managers, and they compare our results to all the other laboratories that also participate in this program, and then give feedback to the laboratories in terms of how you perform in comparison to all the other laboratories. So it's a performance assessment. You're measuring your proficiency at doing these analyses. And these help us to look at our values. If we're off, the bias will show up. If our ability to be accurate in terms of minimizing that variability is off, that shows up as well. And so it's a way just for us to get a third party to give us feedback on how the laboratories are performing. And it's done at least quarterly, quarterly or 3 times a year. We also get known samples from these labs. They're known from these programs. These are plant samples that have been analyzed in this same way across the country. So we can buy the excess of those samples and use those internally for our own quality assurance programs that we practice in all the laboratories. Next slide. So those known samples are, for example, that picture on the right from the ALP. That's a standard reference material that we purchased. We use that then and analyze that periodically, as well as we have our own internal standards that we use that we analyze in every tray that we run so that we know how well we're performing and are we performing to our own expectations. If not, we'll go back and redo the analysis to make sure we can do it right every time. These are methods that are expected of us by the state regulatory agencies, and we also follow methods that are universally accepted by the industry. So they're standard methods, standard procedures, and every laboratory follows those as well as their internal laboratory optimization programs to reduce that variability and to focus on our quality assurance as we go on. Thank you. So that's how laboratories minimize the variability. And Garrett's going to take over now and start talking about his program in terms of what goes on in the field and how to use that information best to manage what's going on in the field and deal with that variability. So thank you very much. And Garrett, go ahead.
Yeah, thank you, Jim. That was a great overview of kind of what we're doing in the lab. And so I want to kind of take this out and take it to more of the field aspect. And when you actually get a report back, what are we looking at, and how am I supposed to interpret this? So when we start interpreting the results that we're getting back, it's a reflection of the whole process that Jim just outlined there. The sampling strategy, the quality of the sample that's pulled. And then when we start looking at the actual reports, it's, there's some assumptions that we're making on ranges, and most of those are general assumptions. So it would just be for your average field, I would say. And so when we start, there's a lot of programs around the country these days that are trying to take yields to the next level. And for those, you're probably going to have to, at some point, come up with your own ranges, but just using our ranges as some sort of a guideline to, we're a little bit low, a little bit high on certain things, and evaluating our ranges throughout the year. So ideally, we want multiple samples taken from the same field, say a good area and a bad area. If you're trying to diagnose something, I have a yellowing spot in the field, something like that. That gives us a good baseline for what a good area in the field is looking like for you, as well as what maybe a problem spot is looking like. And keeping in mind that these samples are just a snapshot in time. So we're not going to be able to solve a year-long problem with one single tissue sample from one bad spot in the field. We can tell you that, yeah, it's looking a little bit low in nitrogen or phosphorus, but to say that we can take one sample and come out there and give you a be-all end-all solution to your problem is probably pie in the sky type stuff. The more we can gather about the field, the easier it is to conclude what's going on in that field. So that's, multiple tissue samples. Maybe that's a sample before and after an application. Maybe that's, I strongly encourage the use of soil sampling when we're tissue sampling, because then it's allowing us to evaluate what we have available to the crop, as well as what's in the crop currently. So, I liken it back to somebody that has diabetes. If you're not going to take a glucose reading at 8 o'clock in the morning and use it for the entire week, we're going to take multiple glucose readings to make sure that we're getting the right amount of insulin. And it's no different for tissue sampling your crops. It might not be every day, but it's looking at it over time and then trying to help make the best decision we possibly can. So a little bit about in-field variability. So this goes back to what I was just talking about. So the blue circle would be like a problem area, and the yellow circle would be what we would consider a good area. And if we just take one from the blue area and make an inference on the whole field about that, we're probably making a bad assumption on the whole field. Whereas if we take one on the yellow, out of the yellow circle, it would be the same thing. We're probably thinking that the whole field is a little bit better than it actually is. So the other things, we're looking at nutrient uptake curves. So each crop is going to have their own nutrient uptake curve. These are readily available from your state universities. And I highly encourage the use of this, so that we'll get into nutrient timing a little bit later, but we want to kind of front end load these crops, because by the time we're determining number of rows around, length of the ear in corn specifically, by the time we get there, if we're just putting enough nutrients on by the time we get there, then we're probably too late, because that plant's already at a disadvantage, doesn't have as much nutrients as it needs, versus It's going to take a little bit of time to get that fertilizer into the plant. So then we're talking about, why should we take a soil sample when we're, you want us to tissue sample and take a soil sample now. And it really comes back to, are you trying to remedy a spot in the field, or are you trying to gain information on a field throughout the year? Things like pH change. I've seen pH change as much as a half a point through just the soil warming up and those biological processes firing up from say a frozen winter to a springtime, and that can open a lot of eyes for people when they see, oh, my grid sample said I had a 6.5 pH, but now I'm down to a 6. Maybe looking at some things kind of outside of the box, I would say. Using CEC to determine how much of, say, potassium is available to my plant versus what is my actual part per million. My part per million might be okay, but if I don't have enough available to the plant, then we can start seeing things like potassium deficiencies in our tissue samples. Crop yield variations, and that comes back to just where we're taking the samples from. I like to use historical yield maps to kind of locate the two spots that I want to use tissue samples from the field, and that's just trying to bring that lower end a little bit higher in the yield, and then maybe push our higher yielding stuff a little bit more. It can help us use our fertilizer. So how efficiently is that fertilizer getting into the plant? Maybe we have different CECs in the good versus bad soil, and it's allowing us to evaluate stuff like that. Is our soil mineralizing or immobilizing our nutrients that we're putting on? Is there an application that can be made based off of that information? And then the forms of fertilizer that we're putting in, is it in the nitrate form, the ammonia form? Is it in season, in the spring? Is it in the fall? Things of that nature can very, very much be affected by what we're seeing in season. So going back to some of the soil testing characteristics of how that affects what's available to the plant, and how that can affect what our tissue values are. We look at say phosphate, for example, and we start to get to some of those lower pHs, and we can see that aluminum and iron are starting to affect our uptake of phosphorus. And so it's things like this that can really, really put that whole picture out there of, hey, my phosphorus is low compared to your ranges on my tissue samples. Why is that? Well, it would be hard to tell you that it was, an aluminum situation if we didn't have that soil test to go with it. And like I said, this is one of the biggest keys to nutrient availability, and some of the applications that you're going to make throughout the year. And this is very much helpful when it's coming to try to remedy those situations. So then getting into CEC a little bit, the main one for this is probably potassium. Just evaluating, if I have a lower CEC and I'm a lower part per million on my potassium, is there a way for us to make in-season applications? Because as we're going to see a little bit later, when we start looking at nutrient uptake and looking at nutrient use versus removal rates, maybe we are at a lower CEC, we're at maybe a little bit lower availability, and we're already starting out with a low part per million. Well, now we can start looking at making some of those in-season applications of potassium, nitrogen, things of that nature. And without it, like I said, without a soil test, we wouldn't really know any of that. We would just be kind of guessing and just saying, hey, our potassium is low in our tissue, but not really getting the full picture. So now we'll move into the role of some of the macronutrients in the plant. And when we're starting to look at those at those tissue values, why is it that nitrogen is one of the most, it's one of the most talked about nutrients probably. It's, as you can see in the diagram on the right, it's right around that. The magnesium is the magnesium molecule is the central nutrient in the chlorophyll atom versus the nitrogen surrounding it. So, it's contributing to that green color. And as you guys know, the nitrogen deficiencies it's interrupting that growth process and starting the yellowing of that decrease in chlorophyll. And then what I really start to look at is what sources are we using when it comes to nitrogen? Are we using ammonia and hoping that it's converting to nitrate and then getting into the plant after that? Or are we applying ammoniacal sources that we know are available right away and getting into the plant, and we can watch those values kind of level off? So when we start evaluating our nitrogen tissue values, and this is in corn specifically, soybeans are a little bit different, but corn specifically, when we take that, so this would be an example of, say, 3 tissue samples throughout the year. So say 1 in June, 1 in July, and 1 in August. That first sample that we take is always going to be the highest percentage of nitrogen in the plant. And as we'll see in the next slide, this is because when we get to that VT time frame, we're using so much nitrogen that we can't possibly put on enough to make that kind of curve go up. The goal is to make it go as flat as possible. So if we're starting at 4.25% here, if we got to August and we were down at 4%, that's kind of the line that you're trying to look for. It's going straight across the paper, not down. And like I said, that's just because of the amount of nitrogen we're using by VT. And a lot of this is just due to how it's being taken up by the plant. And then, as long as we don't get any sort of like large flushing rains, it can really be a tool in managing our nitrogen and just evaluating how much we are actually getting into the plant. So this is some work done by Dr. Flannery, and this is for 308 bushel corn. So these numbers are probably a little bit higher than most people are seeing, but it just gives you the perspective of when we get to VT, we're using 11 pounds of nitrogen for 13 days. So that can be pretty eye-opening to some people. If you're going out and putting all of your ammonia out in the fall, are you just guessing about what's going to be there in the springtime? Say you put out 250 pounds. Are you sure that when you get to VT, you're going to have 225 of that still sitting out there? And you're guessing a little bit. And then looking at across the board, when we get to VT, the phosphorus that we're using, 2.85 pounds a day for 13 days, so around 60 pounds. And then potassium is the other one, and potassium I think gets overlooked a little bit, at least where I'm at specifically, because when we're looking at nutrient removals for potassium, we're only looking at say 92 pounds of potassium getting taken off by this 308 bushel crop. But when we start to look at the nutrient use, you can see by early dent, we're almost using 400 pounds of potassium. And so it gets overlooked a little bit because nobody's going out there making applications based on the nutrient use. We're basing that off of removal. But if we're starting out in low CEC soils, we're starting out with low part per million potassium values, then could it be beneficial to go out there with, say, 50 or 100 pounds of potassium on top of what you might be applying in dry throughout the year and just supplementing this crop and just helping build some of that cell structure. So another tool for nitrogen management is using the crop uptake curve. So this was also done by Dr. Flannery. And it's just showing how much nitrogen is picked up, and they're using growing degree days as their units. And so you can see that you get almost right a little bit before 1,000 GDDs, and it's going almost straight up. So that's about that time of just prior to VT, and we're starting that steep uptake curve of nitrogen. And so do we need all of that nitrogen at say 100 growing degree days, or are we better off applying that closer to say 500 to 1,000 GDDs? And so just looking at nitrogen in general, it's not as stable as other nutrients, and some considerations for, what are you putting on pre-plant? What are you putting on in season? Are you doing both? I think as a whole, ag is moving a little bit farther to the right of that, and getting to some of those in-season applications. And then something else is the depth of your sample. Are you taking profile samples? Are you just taking topsoil? What was the previous crop? Looking at some of like the vegetable crops, you could see those higher nitrate values. Looking at organic matter, looking at the method of nitrogen being applied. Are we applying it 2 by 2? Are we, incorporating it? And then the last thing I would put a plug in for some irrigation water testing, and that can go across the board. That can be in Lincoln, Nebraska, or that could be in somewhere in California. Looking at those nitrates that are coming from that irrigation water can be supplying some of those high nitrate values. So nitrogen deficiency in the plants, like I say, that yellow V coming from the tip along the midrib in corn, and then just yellowing, that's a strawberry crop on the left. So getting into phosphorus, used for storage and transfer of energy, restricted growth and development. So a lot of the times we're going to see decreased root structures. Fruit in this case is smaller and ripening is delayed, altering flavor. Like I said, better root structure with adequate phosphorus and better quality of fruit and improved disease resistance. So you can see on the uptake curve for phosphorus too, you get about to that VT time frame, and we're starting to take up the majority of our phosphate. And so just showing that at VT, we're taking up 2.85 pounds for 13 days. So that's 2.85 pounds every day for 13 days. So 57 pounds by VT. So then reddening of the leaf margins in early corn, and then decreased root structure and just overall plant growth in strawberries. So now on to potassium, giving the crop a good cellular structure, regulating CO2 absorption through the stomata. And then something that I think goes a little bit overlooked for potassium is that it's going to help us in those drought situations. It's pulling that water into the plant roots. In deficiency is reduced production of ATP and CO2 fixation. So we see potassium deficiencies mainly in drought stressed crops, because that there's not that moisture in the soil that's moving towards the roots, and so it's not getting as much of that potassium. So we can see by VT we've already taken up 308 pounds total of potassium, and that's 15 pounds for 13 days. at VT. And so this is where some of those rec concerns come in. Maybe you only have 100 pounds out or 100 part per million in your soil test. Well, do we have enough to get us to this point, or should we think about maybe putting on some, whether it be wide dropped or in season or through the pivot, however you have the ability to do it. So you can see the leaf margin burning in the corn plant. You can tell it's pretty dry there. Reddening in the strawberry plants. In corn, this usually starts in the first 6 weeks after planting. Because of the decreased root system, it hasn't had enough time to develop. So we're not taking in quite as much through that root mass. So getting to fertility considerations, yield goals, we got to be realistic with our yield goals when we're coming up with this stuff. Like I was saying earlier, we're looking at crop requirements, not necessarily saying go out and apply and build your soil levels up to the crop requirements, but just being aware of what the requirements are and having the ability to make an application in season can go a long ways in helping you get on to some additional yield. So the nutrients available in the soil, what are we losing from the soil? Timing of our fertilizer applications we're going to get to next, application methods, and then the economic return. So this is a pretty key one for corn specifically. Soybeans are a little bit different, but these are kind of the top 3 timing windows, I would say, that have been identified. And kind of when I started doing some of this stuff, it was very like, V6, we knew that number of rows were determined. And we looked at it more so like, well, V5, then we'll go out there and put some of our nutrients on. And in time and going through, the amount of tissue samples I've went through, we more so look at it as that corn crop is determining the number of rows around between, it could be even earlier than V3, say V2 to V6, V5. And so when we're thinking about that, we can affect that crop the entire way through that window. It doesn't just have to be backloaded at V6. So if we're trying to affect change over the whole time, we should probably have it at say V2, so that it can be getting mineralized by the soil and then being taken up by the plant while it needs it, not when it needs it, I guess is how you'd say it. And earlier timing is usually better, just because we want to be able to get that nutrient mineralized and into the plant so that we're affecting that. And same thing with the number of kernels long as well. It's getting it in there by VT, not at VT. Because once we've got to VT, a lot of the times it's already too late and we're not going to be able to affect it as much as we would like to see. And I see a lot too of people will make an application of nitrogen at, say V10, and then the next week they'll go out there and pull a tissue sample, and they might have pulled one at, V5, and their percentage of nitrogen is just way lower, and they automatically come back and say that the lab is wrong. But as we've already kind of shown that uptake curve is so great that we can't overcome that. And so we're going to see those values almost tank. So I just wanted to end here with a couple of our different reports. This would be if you just sent in one single sample from a field or from a track, this would be what you would receive back. So you can see we got percent nitrogen, percent phosphorus, potassium, magnesium, all the major major nutrients. And then you can, chloride and molybdenum are two that you would have to request separately, as well as nitrate part per million. And then we also have the ability to track these throughout the year. So say you wanted to do 5, so this is a potato, for example. You wanted to do 5 samples throughout the year to track the crop, and how is that nitrogen or how is that phosphorus moving throughout the year through the plant. This would be the way that you would want to do that. And then also on the graph, we're showing ranges as well. So we also have the ability to put 2 of your crop lines on here. So like if you did take a good and a bad, we would be able to plot those as well. So in summary, the results that you receive are going to be a reflection of the whole process. How are my soil results affecting my tissue sample results, I think is a big one. And just looking at that, like I was talking about earlier, those pH issues, things of that nature. And then letting nutrient uptake curves and growth stages, helping determining your application timing and how you're looking at what's getting in the plant. A lot of people I'm sure are going to be looking at, well, am I too late for some of the stuff that we're doing? And then just considering the removal rates, just knowing that those are very real, and they're very something that not everybody is thinking about, but have a great effect on your crop. So I think with that, Catherine, I am done.
So we did have a few questions come in, and I'm going to try and direct those questions based on when they were submitted and who was speaking at that time. And we had a question come in really towards the beginning of the webinar. So Jim, I guess this would be for you. Do you know of any studies, or do you know if there's a difference between lettuce plant tissue nutrient concentrations and lettuce grown in a hydroponic grow tower with no soil, only water with adamants?
Question. I don't know of any studies. I'm sure there are studies that have looked at that. Just thinking about it a little bit, the advantage of the hydroponic situation is that the nutrients are all there and available in hopefully ideal concentrations. And so it's not affected by characteristics or certain situations that happen in the soil, as Garrett outline, pH changes and CEC and things that diminish the availability of nutrients. In a hydroponic situation, you're in principle supplying everything at optimum rates throughout the growing season. So the uptake should be optimized, and the growth should be optimized given the other edaphic factors or climatic, environmental factors are appropriate too. So I don't know of any specific research, but I would think that the uptake curves and the uptake rates would be optimized in hydroponic in a growth situation.
Great, thank you. Get ready, Garrett, because all the other questions came in while you were talking. Yeah. So do you suggest collecting soil samples at the same time as tissue samples?
Yeah, so it kind of depends on the situation. If I was going to go out there, say like 5 or 6 times throughout the year, I wouldn't take a soil sample every time. I would maybe start with a soil sample, like maybe before you planted, or say if you had a grid sample, you could use that as your starting point. And then going back to some of the uptake curve stuff, prior to VT, then taking another soil sample to see, I can take that soil sample, compare it back to the 2 or 3 tissue samples that I've taken since my last soil sample, and then use that to evaluate, so what we did a lot was use that to evaluate how much like nitrogen we would have available, and then evaluate, do I need to make another application of nitrogen, or do I have enough out there that at this point in time for what my yield goal was for the crop condition, maybe I'm not going to put any more nitrogen on.
Great, thank you. How can crop imagery data be used with a plant tissue testing program to track nutrient availability through the season?
Yeah, that's a really good one. What I would use that kind of information for would be to determine where I would want to take some of those soil samples or some of those tissue samples. So, maybe instead of using a rear facing yield map or a combination of 5 years of yield maps, maybe I'm going to use that imagery for say like a lower NDVI score. Is I'm going to go to that area for my lower yielding, or what I would call a lower yielding block, and then use the greener part of the field as my higher yielding block.
Thank you. Is there a link we could provide for research conducted around the nutrients uptake per day?
Yeah, I can grab that. We can email that out to them.
James, we will send you an email with that information directly from Garrett. Can plant tissue sampling help identify soil management practices which enhance nutrient cycling and increase plant available nutrients within soil?
So yes, there's a couple different train of thoughts I have. My first one would be to come back to some of our cover crop biomass testing. That's not really it might not be exactly what you're talking about, but so what we've done in the past is look at, as that cover crop biomass is breaking down, it's releasing some of that nutrients to the crop. And so can we quantify what that is? So we've put it into pounds on our cover crop biomass reports. Can we see that on the tissue samples from the crop? And I would argue that we can. It takes a lot of work, and there's a lot of groundwork and a lot of stuff that goes into it. But we have done that in the past and seen that and it's not going to be, you're not going to see your nitrogen go up like I was talking about, but what you're going to see is that area is going to stay higher longer than an area of like a different cultural practice, I guess I would say.
I would just add that it's going to be hard to really know that unless you have some comparative values. So, when you're looking at a soil management practice, what was the practice before? What's practice now? If you can compare that, or you have side-by-side comparisons where you have a cover crop in place here and not here, and then what is the management practice doing? But I think what Garrett said too about keeping track of not just the nutrient concentration, but the total nutrient uptake, the total biomass and distribution of those nutrients in that biomass, so that you can assess how much nutrient is being utilized out of the soil, will be very important as well. Good.
Thank you. What is the percentage of plant available nutrients compared to the total nutrients in an average soil sample of silt loam soil?
That's good question. Most phosphorus, for example, the evaluation of phosphorus is used with, we extract the soil with a weak acid, and that number is more like an index of what's available than an actual amount of nutrients that are available for the plant. So if a number like 25 ppm or 20 ppm with a Bray extraction, that index indicates that there's enough there, but what's actually the total amount that will be available, you'd have to do something like we just described, do a total biomass assessment of that field and see how much is taken up in order to really get an idea of that. And that number is much lower than the total amount that's in the soil, because Some of that nutrient is tied up in minerals that are just not available. Some of it's tied up in organic matter that slowly is released, and the organic fraction is always cycling. Some of it's being immobilized, some of it's being mineralized. And so but the percentage of plant available nutrients is always lower than the total amount in the soil, whether it's PK or even nitrogen. Yeah. The soil test gives you an index, and it's a calibrated index, so it's a useful tool, but it's not a total exact number.
Thank you, Jim. We only have a few more questions as long as no more come in. Perhaps you already stated, were the optimum ranges in the results of the tissue analysis based off university sufficiency ranges, or how are those optimum ranges considered?
I'll let Jim, do you know that one off?
The ranges that we have in our reports have been they've been collected over a long period of time. They're published. Some of them are published by universities. Some of them are published by groups of researchers at universities that come together and make a consortium of researchers that say these are the kind of ranges that we should work with. So you can find those in public on public spaces. Generally, what we try to do is base that on that kind of public information as closely as we can. So our numbers will reflect that. They may be more conservative than what an aggressive grower, like Garrett mentioned before. An aggressive grower may want to shoot for 4.5–5% nitrogen in their corn crop at an early growth stage, whereas university recommendations may say 4% is enough, for example. And then also, if you looked at the report, those lines were straight across on those reports. And we do allow for different growth stages. There are different ranges that are appropriate. And we do allow for that in the recommendations when it's one sample at one time. But when we do that summary report, we end up just using the original number and then projecting that number all the way across so that you can see how it compares over time.
Okay, thank you. If high yielding corn needs 200 parts per million K, why are UW optimum recs often much lower than that level?
I would, not being in Wisconsin, but I would probably say just because that's what they've been able to prove to whatever yield goal that has been set. I don't know, Jim, do you have anything else on that?
That's really what drives it is where do they see response? And if you found that you don't get a response over 150 ppm, then they won't recommend more fertilizer above that, other than to maintain that level of fertility. And by response, is it less than 5% chance that you'll see a response to applied fertilizer? That's really what the cutoff is for that high level. Two other things to keep in mind. One other thing particularly, the UW recommendations are based on the Bray potassium extraction, which is not the same as ammonium acetate or malic extraction. Those numbers are usually about 70 to 80% of what we would see with an ammonium acetate extract. So if this 200 ppm is a malic extract, that would be 160, something like that in a Bray extract. So that's one reason why the numbers are different. And then just like Garrett said, the target of where you want to maintain that and where you likely see a response is based on field applications, field research. Incidentally, Iowa just went through a process of updating their recommendations, and what used to be adequate from 160 to 200 for potassium is by malic or ammonium acetate is now 170 to 220. So those ranges did change, and that was just recently because of seeing more response to more fertilizer at higher ppm rate levels. So is that because of the crop? Is that probably because of different management and cropping practices and the plan itself.
Okay,
Okay.
final question, not only because we are actually already over time, but it is our final question we've had submitted. It kind of ties in, Garrett, to one of your responses. Do you feel plant biomass should be taken into account in addition to the nutrient concentration for identifying whether plants are growing well?
Yeah, because I think as we've seen with some groups that are doing a lot of intensive tissue sampling, as that biomass goes up, we see things like nitrogen value percentages go down. And so I think that's definitely part of it. The concentration in the scoop that we're taking is lower, primarily because there's more biomass. I feel like.
Okay, great. As Garrett switches to the next slide, we want to thank everyone for their time today. We will be sending out this recording within the next couple of days for anyone who would like to view it again. Thank you so much to Garrett and Jim for your time today and your expertise. We really appreciate everyone, and we look forward to seeing you in a future webinar.